Yeah, we are prepared, but the problem is the students who are still here and they are not really prepared.
I don't know why they are not prepared. I gave you the notes on Monday or Sunday, so they have the whole week to prepare the card. What are they
Yeah, but
doing?
yeah, they are doing practicals. Morning and afternoon, they are doing their practicals. Biochemistry, microbiology, and anatomy, and even physiology.
And then if you have a card, it not means that you are going to stop the class and study for the card. So card is a continuous assessment. You should attend the class and prepare the card. So you have the night, you have to use the nights for preparing for card preparation. So I don't know why they are saying that they don't have time. So when are going to have time for the card?
Like the next week.
You should adapt to the situation because this is you are going. This is not the university's condition.
No,
So
let's do it on
giving
Monday.
you two, giving you 2 weeks for cut preparation, I think doesn't make sense. So I will
Yeah,
give you
let's do it on Monday lecture.
cut.
Actually, I'm not getting you.
Hmm?
We are not getting you. Your microphone was muted.
Maybe I'm asking if GACO students are attending this class. Are they
No,
some GACO students?
they are in their practicals, all of them.
Okay. And they asked them, the class representative to come to my office. They didn't come. So you have a cut on Monday, right?
Yeah, yeah. Yes,
Yes.
yes.
Get prepared for it. So
You have to do it
get
on
prepared.
Monday.
Yes, okay. When? Morning or afternoon?
Afternoon would be better.
Afternoon
Afternoon.
will be better. Don't you have a practice in the afternoon?
No, we have finished
No.
all those.
So now
But the
you
question,
finished practice.
yeah, we have
Yeah.
finished because but if we do it on Monday, Gakko students will not be available.
Okay. So let's start. So now we are going to talk about the gene regulations or regulation of gene expression. So this gene expression refers to multi-step ultimate kinds of gene expression, or there are two kinds of genes. So those genes are those who are so those who are meeting the basic cellular needs. So those are called housekeeping genes. Generally, those who are continuous produced are called housekeeping gene. But there are other one who are made under certain condition. They are called regulated genes. This can be turned on or turned off. So note that in general, there are genes who are always produced. They are called housekeeping gene. And they are the other one who are made for purpose. So those are called regulatory genes. So these can be either off or on. So the regulation of gene expression or gene control is concerned with all possible ways in which gene activity can be controlled. For example, determining the amount of the products of a particular that need to be produced and the right time to make them. So in prokaryotic cells, I'm going to give you an example of gene regulation in prokaryotic cells. Called operon. So operon is a class of genes sharing a promoter and regulatory sequences. So operon is typically include regulatory gene. This code for DNA binding protein that acts as a repressor. It has a promoter. A promoter is a DNA sequence binding RNA polymerase. It has an operator. Is a portion of DNA repressor bind, and there is a structure gene code for enzyme or protein needed for operator operon metabolic pathway. So the role of operator in a prokaryotic this image here, this figure. So are you seeing this DNA?
Yes.
Okay, good. So in bacteria, glucose for its metabolism. So bacteria needs the glucose as a source of energy. But sometimes the glucose is not present. So there is a lactose.
We are not getting you, lecture.
And you know, the lactose is a combination of what? Network problem. That's why you don't like this struggle of online teaching. So I was saying about, sorry, energy. It has to use glucose. The glucose is the source of energy of bacteria. So in some cases or in some situation, the glucose may not be present, but the lactose is there. And you know that the lactose is made up by galactose and glucose. So this lactose will need to be broken down so that the bacteria can have the glucose to be used. So this is made by a regulation of gene expression by a process called lac opero, so or lactose opero. So how is it made? I'll show you this DNA template. So in the cytoplasm of the bacteria of the cell, the lactose will be there, and it will be called, is what we call the inducer. The lactose is inducer, which is present in the cytoplasm of the cell, or of course, prokaryotic cell. So on this template, there are three regions. One is promoter region, another one is the operator region. In operator region, there is a protein which is always bound there, is called a repressor. So are you seeing here this region?
Yes.
Okay, so a repressor is a protein which is always binded here. So in cytoplasm, there is inducers, which are the lactose. So once the lactose are present in the cytoplasm of the bacteria, which is also called as an inducer, they will come and bind on the repressor. So the repressor, which is found here. Once the inducer is bounded on the repressor, then the repressor will be detached from the operator region of this DNA. So once the repressor is detached, and then the messenger RNA will come and bind the promoter region, and they start the transcription. So by that, in transcription, it will make 3 genes or 3 protein or 3 messenger RNA. The messenger RNA will be coded for 3 genes. So there is what we call RAC A, RAC B, or RAC Y, and RAC Z. So this RAC A is coded for beta-galactosidase enzyme. So this enzyme will be responsible for the breaking down the lactose into two, one galactose and the glucose. So RAC-Y is coded for permease. Permease is the enzyme which is responsible holes or breaking the cytoplasm of the cell so that the lactose can enter the cell. And the LAC-C or LAC-Z is not yet known. So the function of this LAC-C or Y is not yet, or Z, sorry, is not yet known. The Lactose Operon in summary. I think you get it, how it is done. Yes, are we together?
Yes, together, sir.
So you get it?
Not much.
Not much. I said that a bacteria has to use glucose as a source of energy. So in case the glucose is not enough, it may use a lactose. The lactose have to be broken down into 2 parts. One is glucose, another one is galactose. But for this, some protein need to be synthesized, which means need to be translated. So those proteins are beta-galactosidase, and for breaking down lactose into glucose, and permease, which will cleave or in the cytoplasm, the membrane of the cell, so that the lactose can enter the cell. So how is it made? I told you that on the DNA sequence there is regions. One is operator and in the promoter. So the operator region, there is a protein which is called a repressor, is always binded there. So once in the cytoplasm of the bacteria, there is a inducer, which is lactose, it will come and bind to the repressor. So once it's binded to the repressor will be detached, and then the messenger RNA will come and bind it to this site of the promoter site. So once it's bounded to the promoter site, then it will initiate the transcription, which will end up by producing on the LAC-Y, which is LAC-Y is this one, LAC-A is this one, the first one, encoded for beta-galactosidase, the enzyme that I told you. It's broke down lactose into glucose and the galactose, and RAC-Y, which is coded for permease, the enzyme responsible for cleaving the cell, and the LAC-C, which is not yet known. So this is the summary for the Lactose Operon. And then of course, this is the expression, the explanation. So the regulator gene is code for DNA binding protein that acts as a repressor. A promoter is the DNA sequence that binds the RNA polymerase. An operator is a portion of DNA where the active repressor bind. I showed you all of these things.
Yes.
So the role of operator in prokaryotic transcription. So the operator is not bounded by a repressor molecule. RNA polymerase passes over, and if the operator, as I told you, is not bounded by a repressor, so if the repressor is detached, then the RNA polymerase will pass over the operator, reach the protein region, which transcript to messenger RNA. I showed you. I told you, if the repressor is, if the inducer, which is lactose, is binded to a repressor, then the repressor will be detached. So once the repressor is detached, then the RNA or polymerase will start the transcription. So if the repressor molecule is bounded by operator, is bounded to the, if the repressor molecule is bounded to the operator, it means that there is no inducer, there is no lactose to attach to the repressor. Then the polymerase is blocked. It means that the polymerase will not be able to initiate the transcription. However, when the inducer, which is our lactose molecule, is present, it will bind to the repressor and cause the repressor to change the shape so that it's no longer bind to the operator, it will detach. And then when it happens, RNA polymerase can proceed with the transcription. So this is the details of what I was saying. So actually, the lactose operon, or example, in E. coli. E. coli is Escherichia coli. This is a bacteria. I think you know what is Escherichia coli. Is a bacteria which is a normal flora in the intestinal tract, but is a pathogen in the urinary tract. I think you know that 80% of urinary infection are caused by E. coli, Escherichia coli. So hope you know that if you have had, if you learned the microbiology. So the E. coli is one of the key bacteria in microbiology. So we use it in science also. So as I told you, the lactose operon is 3 genes that code the enzyme so that break down the lactose. LAC-Z, so LAC-Z is this one. This is the A one. Is coded for beta-galactosidase, which hydrolyze the lactose to galactose and the glucose. So there is also LAC-Y. LAC-Y is this one, which is called for beta-galactose, which is called for permease that facilitate the movement of lactose into the cells. This will facilitate the movement from outside the cell into the cells. And LAC-A, which is this one, the C1, that's code for thiogalactoside transacetylase. Those physiologic function is unknown. So this is bacteria use glucose as a fuel. So this is what I was saying, that the bacteria will use the glucose as fuel or as source of energy. When the glucose is bounded in lactose, then lacking. I think all of this I have. Let's use this also this for as an example. Are you seeing this image?
Yes.
Yes.
So you are seeing the slide. Okay, good. So this is, I'm going to show you how it is done. So in the absence of lactose, the lactose operon is repressed. The repressor is bound on the operator. This will blocking RNA polymerase. This is called negative regulation. For example, I show you here. This is our DNA template. And this is, I told you there is a promoter region and the operator region. On the operator region, the repressor is bounded there, which means once the repressor is bounded, they will know the transcription not happening because the repressor is still bounded there. I told you that messenger RNA does and start the transcription. But if this repressor is there, the RNA polymerase, which is bounded here, cannot pass here. In the other way,
My lecture,
yes,
seeing your cursor.
you are seeing,
You are not seeing your cursor.
you are not seeing the slide.
Where that's one that you are showing us. You are saying here, but you are not seeing what I told you. Now you are seeing it.
Okay, good.
Now it's clear.
I was saying gene. So we know that the RNA polymerase before start the transcription, it is bind on the promoter region, and the repressor, which is this one, is bounded on the operator. So once there is no lactose in the cytoplasm to touch on the repressor, then the repressor will still binded on the operator, which will block the RNA polymerase to start the. So in another way, if there is lactose in the cytoplasm, are you seeing this here, this lactose here? Once it's present in the cytoplasm, it will come and bind to this repressor. After binding to the repressor will be detached from the operator region. So once the repressor is detached from the operator region, then the RNA polymerase will immediately, RNA polymerase, which is found on the promoter region, will immediately start the transcription. And we ended up by making the enzyme that I told you, beta-galactosidase and the polymerase. So now you're getting it?
Yes.
Murabyumva neza?
Not well.
Not well.
Yes. Start. This.
Hmm?
Yes, you must start this slide.
Hmm? Vous avez?
May summarize this slide.
Again, you need me to summarize the slide again?
Yeah.
Okay. Just try to look at on this DNA template. On the DNA template, there is 3 regions. One is operator. The first one is promoter. Another one is operator. Another one is lactose metabolizing genes. This one. Promoter, operator, and gene transcription unit or lactose metabolizing gene. Sibyo?
Yes.
Noneho nababwiye yuko iyi bacteria yacu ikoresha glucose nka source ya energy. Muri bacteria haba igihe glucose itahagije, bacteria ishobora gukoresha lactose, kandi turabizi ko lactose igizwe na glucose na galactose. So kugira ngo glucose iboneke hazabaho galactose izacikwamo ibice bibiri igatanga galactose na glucose, sibyo? Ukuntu bizagenda rero igihe lactose izaba iri muri cytoplasm, lactose iraza tuyita inducer. Hano hari ikintu kiba kibindinze gifashe kuri operator region bakita repressor, ni protein ibiri kuri operator region igihe nta lactose ihari mu buzima busanzwe. Kuri promoter region harimo ikintu bita repressor. Repressor ni protein ibifashe kuri promoter region. Noneho RNA polymerase yo iba hano promoter region. Ntabwo ashobora gukora transcription igihe repressor igifashe kuri operator region, kuko urabona operator region iri imbere ya promoter region. So igifasheho hano rero ntabwo ishobora gukora transcription ngo ikomeze ikore elongation hano. Turabyumva?
Yes.
Noneho once muri cytoplasm harimo lactose, ariko kano muri kubona hano kano gasa na mauve kazaza gafate kuri repressor region. Murabibona isibindinga kuri repressor. Nimara gufata kuri repressor izahita ivaho, izahita ihindura structure then ibe detached, ive kuri ya template yacu ya DNA. Nimara kuvaho bizahita bituma RNA polymerase yari hano kuri promoter region ibona umwanya wo gukora noneho transcription. Ihita izikora elongation hano ikora transcription iza ya lactose metabolic genes zimwe eshatu tuzi, harimo LAC-Y, harimo LAC-Z na LAC-A. LAC-Z tuzi yuko iba ifite beta galactosidase, ni enzyme izaba iri responsible for making iyo enzyme ifata ya galactose yacu iri muri cytoplasm ikayigabanyamo ibice bibiri, ni ituma habaho galactose na glucose. Hakaba indi enzyme yitwa polymerase. Iyi enzyme yo ni ituma lactose iva hanze ya cells ikayinjiza muri cells. Noneho iyindi yo ntabwo na n'ubu ntabwo iramenyekana, science ntabwo ziramenyekana. So yitwa negative regulation igihe nta galactose ihari, repressor ikagumya ibindinze kuri operator, ikitwa positive regulation igihe in cytoplasm harimo lactose ariyo twita inducer, izaza igafata kuri repressor ikavaho, then RNA polymerase ibi kuri promoter igatangira the elongation igakora zira protein. Noneho murabyumvise?
Yes, birumvikanye.
Okay, good. So it is not only always this repressor which is governing the gene regulation in bacteria, but there also cyclic adenosine monophosphate. So this availability of glucose govern expression of lactose digesting genes via catabolism of repression. This will be mediated by cyclic adenosine monophosphate, which will bind to active protein. In the presence of glucose, the adenyl cyclase is activated, and the sufficient quantity of cyclic adenosine monophosphate are made and binded to catabolically active protein. So this binding or this complex will cause RNA polymerase to more efficiently initiate transcription at the promoter site. This is also another way. So once adenyl cyclase is activated, in the absence of glucose, of course, this cyclic adenosine monophosphate, we bind to the catapolite activator operator. This binding. So the binding will allow the RNA polymerase to start the transcription or to initiate the transcription. Nazo zizaza kuri za ntuza twavugaga za RAC gene, REC Y and REC Z, RAC Y and RAC A. These bindings also help in regulation. Okay, this was for lactose. And then for tryptophan is somehow same with the lactose, but this tryptophan I'm going to give it to you as assignment. Is that okay? Is that okay? Are you getting me?
Yes, we are getting it.
So I think you understand about the lactose, and this tryptophan is somehow similar to the lactose, but this one I'm going to give it to you as assignment, so you will make it as assignment.
It's okay, lecture.
Okay, good.
Will we submit it or we know it or not?
You submit it, and I will mark it. So the gene regulation in eukaryotes. Now, let's talk about gene. That one we were talking about gene regulation in prokaryotes. Now we are going to talk about gene regulation in eukaryotes. So there are numbers of ways that gene regulation in eukaryotes differ from gene regulation in prokaryotes. So in eukaryote, the genes are not organized into operons. So in eukaryote, there is no story of operons. Eukaryote regulation genes are not usually linked to the gene in the regulate. Some regulatory protein must ultimately be compromised in nucleus. So in the second begin in the cell membrane. And so this in a gene, in eukaryotics, it's happening in a nucleus, but the signal starts from the cell membrane to the cytoplasm. So eukaryotic DNA is much larger, wrap and around, and we give you something movies. So near all the cells and organisms contain identical set of DNA. However, these genes are expressed in high tissue in some manner. Some genes are silenced, and some genes are silenced or grouped, or one group of cells were activated in the other. So as I told you, there are some regulated genes and continuous genes. So the regulated genes they may be silenced. I told you they may be off or on. So some genes are silenced in one group of cells were activated in another group of cells. For example, most of pancreatic cells synthesize and secrete the digestive enzyme, but pancreatic cells synthesize insulin and glucagon instead, which means that those digestive enzymes were already silenced. So DNA has expressed or non-expressed sequences. So yes.
So you are leading at a high speed that you are not able to get the subject.
I'm just reading, then after I will explain. So I'm just going through the slides, so and then I will explain. I was saying that some genes may be silenced, or for example, pancreatic is responsible for secreting digestive enzymes. But we know that the pancreas synthesize insulin and glucagon instead of digestive enzymes. Why? It's because the genes responsible for the synthesizing or producing digestive enzyme in the pancreas were silenced. Ni ukuvuga ngo hari amagenes akorwa en general amwe, hari n'andi akorwa bijyanye na purpose nabibabwiye, zimwe zita continuous genes, indi zikita regulated genes. Izikorwa rero haba izo regulated zishobora kuba silenced, ni ukuvuga ngo igihe cya transcription zigahagarikwa. Kimwe izi zihagaritswe rero nko kuri pancreas, aho kugira ngo iba ifite ibintu byinshi ushobora gukora digestive enzyme, ariko impamvu itazikora ni uko rimwe na rimwe zimwe ziba silenced. So DNA has expressed and non-expressed sequence. Non-expressed DNA is typically higher considered a form of heterochromatin. So DNA has expressed and non-expressed sequence. Non-expressed DNA is typically known as or considered as known as heterochromatin, the non-expressed one. So the transcriptional active DNA, which is known as a chromatin, is less considered and presumptively provide access to transcription machinery. The transcription machinery gain access to DNA package and chromosome and via chromatin remodeling or histone modification. So example here, there is the heterochromatin. They are those who are silenced. I told you the silenced one, they are one who are there but they will not be transcribed. For those digestive enzymes in the pancreas that I told you, they are silenced. But they are also the activated one, which is called a chromatin, this part. So I don't know if you are not seeing, if you are seeing here. So this
We are seeing there.
So the echochromatin, they are the one who are active. Other one are silenced. So in the pancreas, this is the heterochromatin, the silenced, the digestive, and the glucagon and the insulin, the active one. They are the one who will be transcribed. Okay. So this is chromatin remodeling. It's the process of the enzyme assisted in process of asserting access to nucleotides, remodeling structure, composition and position of nucleotide, creating nucleosome DNA of gene. DNA, this is the notes. So the chromatin contain a non-histone protein attached to also contribute to remodeling. So those who are containing the non-histone protein, they need to be remodeled. Most of common of include the high mobility groups, so named because of the high electrophoretic mobilities in polyacrylamide gel. Polyacrylamide gel. So they have some high mobility groups. When we do electrophoresis in polyacrylamide gel, then they will be highly electrophoretic mobilities. So this is a kind of Western blotting technique. So you don't know it. I will show it to you once we come out on the techniques. So we use polyacrylamide gel. This polyacrylamide gel is sometimes used during PCR or Western blotting. So it's found in laboratory. Could you give me please 1 minute? Then I connect my laptop. Okay.
No problem.
Okay.
Yes. Good food.
Include the high mobility groups. I told you about the polycramide gel. So this is not focus. So histone modification. So the histone modification is catalyzed by this enzyme. So it is the covalent histone modification complex which catalyzes the additional or removal of various chemical elements on histone primary. So the enzyme modification, so they are acetylation. So for this to be modified, so they should be in acetylation, methylation, and phosphorylation. So they are in 3 ways. So one is acetylation, methylation, and phosphorylation. Okay. So see here, this is the methylation on the active form of histone. So it will be acylated on the histone tail, and then it will also maybe methylated, and then finally it's also maybe phosphorated. There are 3 ways of histone modification. So the transcription control, galactose metabolism in yeast. This is going also to be another part of the assignment. So RNA processing control. So in eukaryote, as I told you, the gene regulation, messenger RNA undergoes several modifications before it's exported from nucleus to the cytoplasm for protein synthesis, of course. So we have learned about camping at the 5 end and proadenyl at the 3 end and splicing. So we have learned all the 3 RNA modification. You remember the capping, the polytail modification, and splicing.
So in addition, the messenger stability also affects gene expression. So now we are going to talk about the small RNA in the control of genes. So the small RNA or microRNA. So the microRNA, these genes also are found in a variety of locations in the genome, including the introns of expressed genes. They are also often clustered in multiple microRNA single transcription unit. So I don't know if you remember, we talked about the splicing during RNA modification. You remember it? So we've learned about the introns and the exon. So we have seen that the exon are the coding region of RNA exons. And you also have learned that the intron are non-coding of RNA. And you have seen that during the splicing, the introns are removed. Hope you remember that. So those non-coding region actually is not, many people will say that the non-coding region, the introns, are not necessary. But for you as the great scientist of tomorrow, you have to know that the non-coding region, they have a very important role, even though they are not non-coding region, even though they are called the introns, the non-coding sequences. So these one, they are the one who are coded for microRNAs. So they may be microRNAs, the silencing RNA. So they have three sets of function. So they can make microRNA as well, as I said. So this microRNA, there is a small set of RNA, is a small template of RNA, which is made by 22 nucleotides that process and bind another protein in cytoplasm for the complex that function as replace or express gene based on the sequence complementary to microRNA. So I told you about the microRNA. So the complementary region is usually 3' and a translated region of gene, and results can be cleaved into microRNA or inhibition of a translation. So this microRNA, it's the one who will direct or who will orchestrate the transcription, either by any promotion. So this microRNA is synthesized in nucleus by the enzyme called DROSHA, and then it will be it will go to the cytoplasm for the transcription, for the translation by another enzyme called DISA. So in nucleus is called pre-microRNA, where it will get mature in cytoplasm, of course, by the help of the DISA, and it will now be called the mature microRNA. So small interfering RNAs or silencing RNA. So we've learned about microRNA. That's our intron may code for microRNA. This may also code for silencing RNA or small interfering RNA. So the production of small interfering RNAs is similar to the microRNAs. So the production is somehow similar from the nucleus by the DROSHA, and then it's passed through the new cytoplasm and processed by the DISA. So except that it arises from long pieces of double-stranded RNA. So this RNA, it has to be double-stranded. Like it, then it looks like a double strand. So this is either very long region of self-complementary or to form the complementary RNAs. So it may be one long, which can make a like double strand, or two strand of RNA, single strand of RNA, which will look like double stranded RNA. Then by a process by specific endocrines, and they yield multiple silencing RNA that are loaded into. So this long one, it's one which will produce, the long double-stranded are processed by specific endonuclease. Endonuclease is an enzyme which will lead with multiple silencing RNAs that are loaded into other protein to form a complex and bind microRNA to silence, messenger RNA to silence it in transcription. I give you an example. I told you about how this silencing RNA are produced. You know, there is double strand, and then with the help of this enzyme, it will cleave and give multiple silencing RNA. I said about that. So how it works, this silencing RNA, it works by silencing some sequences which will lead to something else, to a protein that we don't know, or which will enable the cell to make the protein. I told you, I've said that during our previous slide when I was talking about the echo and the one that I told you. About the chromatin and so on. So remember the example I gave you about the pancreas, insulin, and digestive enzymes. This is mediated by silencing RNA. So the source of double-stranded RNA to produce silencing RNA can be either from a cell or outside the cell. Let's have example. Let's give you an example, an easy example. This silencing RNA in the lab, it's used to prevent the transcription of a virus. So once the virus is into the cell, it will integrate its RNA. So its RNA, it may be transcribed and for our body. But the silencing RNA is there to silence the viral RNA. That's why here they are saying the double strand it produces, it can be either from the cell or outside the cell. So outside the cell, it may come as a virus. In the cell, it may be proper RNA. From the cell itself, the gene can produce RNAs which long region of self-complemented that fold back to produce substrate for nucleus as substrate. Exogenous double-strand RNA can be introduced experimentally by infection or viruses. Let's call this silencing RNA as the cell protector. Like for example, let's say call it as a soldier for the cell. In case there is integration of viral RNA, then this silencing RNA will immediately silence. We immediately go to that RNA and make a silence it. In normal ways, like a cell RNA, it may make like a transcription of protein which are not needed. This also may silence them. So I think now you understand what is silencing RNA and what is microRNA. So this is the splicing that we've learned. This is the messenger RNA transport and the control. So this one, you know it. This is a messenger RNA degradation control. So how it will be degraded. So now we have two assignments, right? One is the tryptophan operon. Another one is the galactose metabolism in the yeast. This is not necessary. The galactose metabolism is not necessary. So let's talk about the practical component of this module. So actually, in molecular biology, the basic techniques are PCR, which is a polymerase chain reaction, and for this PCR is for RNA and the DNA amplification. And there is also the Western blotting, which is for protein. So are you getting me? Are you getting me, class?
Yes.
Okay. So now we are going we finished about the theoretical part of the module of molecular biology module. And now we are going to talk a little bit about the practical component. So we've studied about the RNA and the DNA and the protein in theory. So how these are processed in practice? How these are done in practice? So we know that everything have RNA and DNA. And we know that RNA is there for making a protein. So how are they make? How in the laboratory we detect the RNA? How we detect the DNA? How we amplify it? So that's why we are going to talk about the PCR. Have you heard about PCR? I think you all know PCR. The corona, the test always PCR. They ask PCR if you want to travel. They ask PCR if you want to go to I don't know. They ask your PCR results. So the PCR results is this one. The PCR means polymerase chain reaction. So it means it's the amplification of RNA or DNA, amplification of DNA, of course, to make a billion of copies of DNA so that the machine can detect if that
We are not getting you.
Lecturer, you are muted.